In this video, we're going to be taking a look at this beautiful sloth, but also we're going to be taking a look at the factors that affect enzymes. So enzymes are little pieces of machinery inside of our body. They are like Bob the Builder. They're responsible for making and breaking things inside our body, making new things that our body can use, making sure that we have everything that we need to survive, okay? They're the little builders of our body, okay? And they don't always work that well. Just like Bob the Builder. Bob the Builder needs to have all the food and all the right conditions before he can work as well as possible. The same way, your enzymes need good conditions. They can't work well all the time. So which things affect enzymes? Which factors? There are three important ones that you need to know, and they are temperature, pH, and the amount of substrate. Don't worry, all of these three will make perfect sense in this video. So let's get into it. So here we have an enzyme, right? This enzyme has a specific shape to do a specific function. Just like Bob the Builder has a specific tool for a specific function, your enzyme has a specific structure for a specific function. Now, we know your enzyme is going to bind to some things called substrates, right? So these are going to be the substrates. And these substrates are going to bind or collide with the active site. So this is the active site, because this is the area where the action happens. Once these substrates binds with the enzyme can now make a reaction happen. Okay, so it can make a product. So now we can see this enzyme was like a little tool. It put these two substrates together to form a new product. Now, this new product will have a unique function. For example, Bob the Builder might build a little house, and that the function of that product or that house is to keep you warm or keep you safe, right? So just the same way, these new products made by these enzymes in your body will all have different, a diverse array of functions to keep you alive, okay? Now, let's get into the factors affecting it. We'll focus on the first one, which is temperature. This one is super interesting. So here you can see I have a little graph, okay, showing how as temperature increases, so coldest, medium, and hot, and then rate of reaction, right? So we can see here there's an interesting curve. We notice at cold temperatures, the rate of reaction is low. At very high temperatures, the rate of reaction is low, right? But right in the middle, the sweet spot, there's a peak or an optimum temperature where the rate of reaction of the enzyme, meaning the temperature at which the enzyme works the best, it has the highest rate of function, is typically at a medium temperature. Now, why is this? Let me show you. First, I can tell you a real-life analogy. When you go outside and it's really cold, let's say Bob the Builder goes outside and it's really cold. Can you imagine him trying to build something? Have you ever tried moving your fingers really fast outside when it's really cold? It really moves so slowly, right? It moves so slowly. You can't do that much, right? You have to be so slow, okay? So let me show you this. But when it's really warm or really nice, if it's really nice temperature, your fingers move around nice and freely. And then when it's really hot, your fingers are like sweaty and everything's slipping and all this, okay? So let me show you in terms of the enzymes what I mean. So here we have a beautiful cell, okay? So this right here is a cell. I'm not putting in any organelles or anything like that, just for explaining this purpose, okay? So I know there's no organelles, but just whatever. So this is our beautiful cell, and it was an accident that it happens to look a bit like a smiley face. Okay, that was actually an accident. Now, let's think about temperature. So, temperature works like this. The higher the temperature, like I just said, the faster your fingers could move, right? At very low temperatures, your fingers could barely move. So temperature makes, the higher the temperature, the more kinetic energy, meaning the faster these substrates are going to move around, just like your fingers. So at a low temperature, your fingers move slowly. So just like that, at a low temperature, these substrates are going to move around so slowly. They're going to bounce off and move slowly and move slowly. And so eventually they'll get into this active site and bind. Okay? So at slow temperature, the substrate moves so slowly that the interaction took so long to happen. Oh, now, as we increase the temperature, these molecules move fast, and they get into contact with this thing, the active site, really fast. So they move really, and then they bounce everywhere, and then they land in the right spot. Okay? So that's at a good temperature, because temperature increases movement. Just like your fingers. When it's nice temperature outside, you can move your fingers really freely, right? Now you might be like, okay, that sounds awesome, so the higher the temperature, the better. That's not true. When it gets too high, something bad happens. So notice how I said with high temperature, things start moving fast, right? We gotta bear in mind, remember that this protein, let me show you. Actually, no, I had it down here. This protein is actually made up of, this enzyme is actually made up of proteins, right? Proteins are like basically long chains of these little balls called amino acids. Okay, so an enzyme is just a protein. Now, if it gets really warm, remember how these things move really fast. The same way, when it gets really warm, these bonds start to move, and the internal structure of this enzyme is going to change because these molecules are going to move around, and the enzyme is going to change shape. And now, if the enzyme changes shape, then these substrates may not fit anymore. Let me show you. So instead of having this beautiful functioning enzyme, we may have something like this now. Okay, you can see this enzyme kind of melted. The temperature got so high that the protein that this enzyme is made of started deforming. It started moving around so much that the enzyme is now a weird shape. And now these substrates, look, they don't fit perfectly, right? They actually don't, which means that the product cannot be formed because they gotta fit like lock and key, remember? So does this make sense, right? So now at high temperatures, the rate of reaction is actually very low. Okay, it just breaks the enzyme. The enzyme gets denatured. This is called denature. You can see this. A good real-life example is with an egg. At first, when you put an egg into a pan, it is transparent, right? The egg white is transparent, but as soon as you start boiling it, it becomes white. That's because the egg white is made up of protein, and this protein got denatured. It got changed in structure, and as the structure changed, now it became white. Okay, so that's a real-life example of it. So hopefully this one makes sense. Too high temperature, it just breaks. And very low temperature, it's very slow, and it takes some temperature to start working, and then suddenly it reaches a sweet spot and works really well. Okay, so I hope this one makes sense. Now, next one. We get to pH. This is a trickier one, okay, so bear with me. So here we have, again, the beautiful cell, but the graph looks different. Notice, I'm putting pictures here to make it interesting, but on this side it's low pH, and at this side it's high pH. The lemon here represents acidic, and this one represents basic, and the water represents neutral, so low to high pH. Now, we can notice that the pH has a major effect. At a specific pH, the rate of reaction is very high, but then either side of that pH, the rate of reaction decreases a lot. Now, interestingly about this curve is this curve can depend. It could be here, it could be here, it could be here. It depends on where the enzyme is. Okay, and I'm going to explain that further. Let me first show you why pH matters. So pH is basically determined by the ratio of H plus and OH minus. Pay attention to this. H plus, so positive charge, and OH minus. The more OH plus in your body or in your specific cell or wherever we're considering, the more OH plus, the more basic. The more H plus, the more acidic. So a lemon is acidic, meaning it has more H plus than OH minus. Okay, so acidity or pH is determined by these two molecules. So let me show you why then water is neutral. So water has this structure, H2O, right? And water is consistently either in this shape or it is broken down into these two. So you can see the OH comes from here, OH, and then the H comes from there. But when they break apart, they have these charges, OH negative and H plus, okay? So that's why water is neutral, because when water breaks, it forms an even amount of this base and an even amount of this acid, and hence they cancel each other out, and we have a neutral, or a pH 7. pH 7 is the neutral number, right? Now, some molecules, so I'm not going to show it here, but some molecules form especially a lot of H+, or especially a lot of OH+. They don't have this balance. And when you form more H+ than H minus, then you're going to be acidic. And if you form more, if this molecule forms more OH negative than H+, it's going to be basic. Okay? So, why does this matter? How does being very acidic or very basic influence how an enzyme works? Basically, get it? Basically. So, essentially, the enzyme, remember how I said it's made up of all these proteins, right? These proteins. These proteins have little charges on them. So for example, let's say this spot of the enzyme is a positively charged amino acid, and here it's negatively, and then here it's positively. If you have too many of these H pluses and OH negatives, so let's say we have a molecule that has a lot of H plus for some reason, okay? A place in the body like the stomach. The stomach is very acidic, right? So let's say we're in the stomach and we have a lot of H plus. If there's a lot of H plus, it's going to be acidic. And the reason why this makes some enzymes not work too good is because these H pluses will go and interact with the negative. Remember, positives attract. It will go interact with this negative charge on the enzyme. And this means that the substrate can't fit anymore. The H plus is blocking it. And hence, so at too high of an acidity, or if it's too acidic, this could influence it in this way, and now the enzyme can't work. Vice versa as well. So for example, say it's too basic. So for example, say in your intestines, it's like pH 8. Or in the stomach, it was pH 2. So in the intestines, it's like pH 8. It's really basic. Now these negative charges, now this OH will interact with all the positive charges, and it will block these sites. Now, these substrates once again can't interact properly because they're being blocked, and so the enzyme doesn't work. Do you understand this? So, what do I mean by the fact that this curve will shift? So let's say we have an enzyme that works in the stomach. For this enzyme to work well in the stomach, what must it be able to tolerate? It must be able to tolerate H+, right? Because it's very acidic in the stomach. And so this enzyme, which is meant to work well in the stomach, will have the curve shift like this to the left, so that the optimal pH for it to work is in acidic conditions. Okay, right? This makes sense. So if you have an enzyme that works the best at acidic conditions, it will mean it will function really well in the stomach because it's always acidic there. Okay, so that's what I mean. It depends on the place. Different enzymes have different response to pH depending on where they work. So for example, another one. In the intestine, like I said, it's very, let me put this picture here. So in the stomach, it will be the pH, the enzyme will have a graph like low because they work well in the acidic conditions. Whereas in the intestines, it will be very high. They will work at a very high pH because there's a lot of OH. So you want an enzyme that can tolerate a lot of these OHs and work well in a high pH. Okay? Now, sometimes in the cell, for example, let me move this. One last example. This is the most confusing one, but it should make sense. So now the last example would be inside cells. Inside cells, typically the pH is 7. And so you want a type of enzyme that can function the best or have the optimum rate of reaction in the pH of 7. Okay? So I hope this makes somewhat sense. So just know that this curve depends on which enzyme we're considering. Different enzymes work better at different pHs depending on where they are. Okay? So if we put a stomach cell, a stomach enzyme in the intestine, will it work? No way. Because look at this curve. This curve doesn't even work at pH 7. So imagine pH 9 or 8. Okay? So an enzyme only works well at the right place. So I hope this one makes sense. Let's move on to the last one. Substrate concentration. So notice this one looks different from the ones we just covered. We have an increase in substrate concentration and still rate of reaction. So here we have a little bit of substrate, here we have more, and then most. Notice as we increase the substrate concentration, there's an increase in rate of reaction, right? That makes sense, and I'll show you why. But then as it gets too high of substrate concentration, the rate kind of plateaus. It doesn't go faster anymore. Why is this? So let's use this picture. So here we have an enzyme. This enzyme can work really fast, okay? So for example, it can take new substrates every 1 second, okay? So let's say here comes a substrate, they bind, product is made, finished. That took 1 second. And now let's say 10 seconds later, another pair comes. Okay, so in that 9 seconds, it had the ability to do more work, but there was no substrate. Okay, so this shows that at low substrate concentration, the rate will be low. But say we gave it some substrate every second, then the rate will be really high, because then in 10 seconds, it can make 10 products. Okay, but so I hope this makes sense so far. But if we give it too much substrate, so we get to a point where the substrate concentration is so high, we may then that substrate concentration may be so much that it's too much for the enzyme to work with. It's like giving one person too much work to handle. No matter, so for example, let's say you have a scissor and you need to cut out a shape, and your friend has to bring you some paper. And for example, let's say it takes you a very long time to cut out the shape. If he keeps bringing you new paper the whole time, that doesn't mean you're going to cut faster, right? Because you are the rate limiting step. You take the longest. So that's the same thing here. If we keep bringing it new substrates the whole time, even though it's already full, it's already occupied, then that's not going to increase the rate anymore because it's already busy. You can't make it work any faster, right? So that's why it plateaus out like this, okay? Eventually, it gets very saturated and very occupied that even more substrates won't help. So that's it. That's the only 3 things you need to know that affects enzyme function. And at the end of the day, that's why if our body temperature gets too high, we will die because our enzymes will stop working and we will therefore not be able to make the right proteins or products anymore to survive. If our body gets too acidic, then we will die too, or too basic. Okay, so that's why these factors really matter, okay, for our body to function at its best.